Dear students, in yesterday class we have learnt about synthesis of insulin through rDNA technology. We have studied. Okay. Today's topic is gene therapy. In human beings, hundreds of genetic diseases are identified by the scientist. Genetic disease means these diseases are heritable one. That is, the disease is passed from parents to offspring. Such diseases are called genetic diseases. You have studied in genetic topics about the diseases. There is no drug for these diseases. Okay. Now we shall see gene therapy. What is gene therapy? Gene therapy means it is an attempt to correct hereditary diseases like cystic fibrosis, a genetic disease, muscular dystrophy, another genetic disease, ADA deficiency, hemophilia, etc. These diseases are due to the defect in the gene. Muscular dystrophy. This is a recessive disease in which the muscles they won't function or disintegrate. ADA deficiency, adenosine deaminase deficiency. The enzyme is not synthesized. This is also called SCID. SCID. Severe combined immunodeficiency. A person suffering from this disease will not have the immunity. It is a dreadful disease. Hemophilia. You have already studied about hemophilia. It is a sex-linked disease. The person who suffers from hemophilia, blood will not clot because lack of clotting proteins. Hemophilia is different types. Hemophilia A, hemophilia B, like that. Okay. So like this, hundreds of diseases are identified by the scientists. Okay. Then, these diseases are not curable diseases. See, any disease bandre, if we take drug or antibiotic, the disease will be cured. But these diseases, that is genetic diseases, are not curable because they are hereditary one. Okay. Next. Then, what is gene therapy? Gene therapy may be defined as a collection of methods that allows correction of a gene defect in a child or in an embryo. So gene therapy means it is one of the method in which the defect genes are corrected in a child or in an embryo. Then it becomes permanent cure. Gene therapy is the utmost treatment of 21st century. Research is going on throughout the world about different diseases. Gene therapy. Okay. In gene therapy, genes are inserted into the cells or into the tissues of a person to treat a disease. It means in this method, the right type of genes are inserted into the cells or tissues of person. Okay, we shall see one such attempt of gene therapy. The first clinical gene therapy was conducted in the year 1990 to a 4-year-old girl to treat adenosine deaminase enzyme deficiency, that is ADA deficiency. It's also called SCID, severe combined immunodeficiency disease. Okay, this enzyme, adenosine deaminase enzyme, is necessary It is very important enzyme for the immunity. This disorder is due to deletion or loss of a gene for the enzyme. Why this disease appears in? Because of deletion. Because of deletion. You know the meaning of deletion? Loss of a chromosomal segment is called deletion. ಕ್ರೋಮೋಸೋಮಿನ ಒಂದು ಪಾರ್ಟು ಇದ್ದಂಗಾದರೆ ಅದನ್ನು ಡಿಲೀಷನ್ ಅಂತ ಕರಿತೀವಿ. ಇಟ್ಸ್ ಎ ಟೈಪ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮಲ್ ಡಿಸಾರ್ಡರ್ಸ್. Chromosomal what you call aberration we call. Deletion is a type of chromosomal aberration in which the segment of the chromosome is lost. It means the gene is lost. The enzyme synthesizing gene is lost. Hence, the enzyme is not synthesized. Okay. It leads to the disease. The persons who are suffering from this disease will not have immunity. Okay. Then, how this gene is treated in modern science? This gene is treated. So this disease is treated or cured by two methods. One method is by bone marrow transplantation, and second method by enzyme replacement therapy by injection. Okay. So now we shall see the steps of gene therapy. It's very important for the examination. Explain the steps of gene therapy. As a first step, lymphocytes are obtained from the blood of patient. That is from the bone marrow, and are grown. Grown means cultured outside the body, that is in the lab. This type of culture we call vitro culture, in vitro culture. Once again, as a first step, the lymphocytes are obtained from the bone marrow, of patient. And the cells are grown or cultured in vitro, that is in the lab. The second step, then the functional ADA, cDNA gene is introduced into the lymphocytes using retrovirus. The retrovirus acts as vector. ಈ ಸ್ಟೆಪ್ನಲ್ಲಿ ADA ಜೀನನ್ನು ಲ್ಯಾಬಲ್ಲಿ ತಯಾರು ಮಾಡ್ತೀವಿ. Then the gene is introduced into the lymphocytes. ಹೇಗೆ? ರಿಟ್ರೋವೈರಸ್ ಉಪಯೋಗಿಸ್ಕೊಂಡು ಜೀನನ್ನು ಹಾಕ್ತೀವಿ. Okay, the second step. Next, third step. Since these cells are not immortal, patient require periodic infusion of genetically engineered lymphocytes. ಈಗ ನಾವು ಆಗ್ತಕ್ಕಂಥ ಸೆಲ್ಸು immortal cells ಅಲ್ಲ. Immortal cells means they can't divide again and again. That is why periodic infusion of genetically engineered lymphocytes necessary. ಹೀಗೆ DNA cDNA ಜೀನನ್ನು ಹಾಕಿದಾಗ ಆ ಜೀನ್ ಇರ್ತಕ್ಕಂಥ ಸೆಲ್ಸ್ನ ಜೆನೆಟಿಕಲಿ ಇಂಜಿನಿಯರ್ಡ್ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಸೆಲ್ಸು ಇಮ್ಮಾರ್ಟಲ್ ಅವು, ಮತ್ತೆ ಡಿವೈಡ್ ಆಗೋದಿಲ್ಲ ಅವು. ಹಾಗಾಗಿ ಮತ್ತೆ ಪದೇ ಪದೇ ಜೆನೆಟಿಕಲಿ ಇಂಜಿನಿಯರ್ಡ್ ಸೆಲ್ಸನ್ನು ಹಾಕಬೇಕಾಗುತ್ತೆ. ಓಕೆ. If the gene therapy is conducted at embryonic stage, it could be a permanent cure. Okay. Both the steps are not completely curative. ನಿಮಗೆ 2 ಸ್ಟೆಪ್ಪನ್ನು ಹೇಳಿದ್ದೀನಿ. ಬೋನ್ ಮ್ಯಾರೋ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಷನ್ ಆ್ಯಂಡ್ ರಿಪ್ಲೇಸ್ಮೆಂಟ್ ಆಫ್ enzyme by injection. They are not completely curative. The reason is cells are not immortal. Okay. Then, types of gene therapy. Coming to the types of gene therapy, there are 2 types of gene therapy. One is somatic gene therapy and another is germline gene therapy. In somatic gene therapy, the gene therapy is conducted in the body cells or in the somatic cells. They are not heritable. It is allowed. Germline gene therapy is not allowed because it is conducted in a germ cell. Germ cells means in sperms and ova. Okay. So that's all about gene therapy. It is for 3 or 5 marks. Even for 2 or 1 mark also there. With this, gene therapy is completed. Okay. Next topic is molecular diagnosis. Okay. As the name indicates, molecular diagnosis, we are making use of molecules. For effective treatment of a disease, early diagnosis of disease is necessary. And also, knowledge of pathopathology of a pathogen is very important. What is the meaning of this sentence? For effective treatment of the disease, early diagnosis is necessary. ಕಾಯಿಲೆ ಬೇಗ ವಾಸಿ ಆಗಬೇಕಂತಂದರೆ ಬೇಗ ಬೇಗ ಕಾಯಿಲೆನ ಕಂಡುಹಿಡಿದಿರಬೇಕು, ಮುಂಚೆನೇ ಕಂಡುಹಿಡಿದಿರಬೇಕು. ಜೊತೆಗೆ ಪ್ಯಾಥೋಫಿಸಿಯಾಲಜಿ ಆಫ್ ಎ ಪ್ಯಾಥೋಜನ್, ಪ್ಯಾಥೋಜನ್ನು ಯಾವ ರೀತಿ ಇದೆ, ಅದರ ಫಿಸಿಯಾಲಜಿ ಏನಂತ ಗೊತ್ತಿರಬೇಕು. ಆಗ ಬೇಗ ಡಿಸೀಸನ್ನು ಕ್ಯೂರ್ ಮಾಡ್ಬೋದು. ಓಕೆ. In conventional method of diagnosis of disease, we are following the conventional methods. Our doctors are following the conventional method of diagnosing the disease. In this method, the disease is identified by testing serum. Serum means blood. Or by testing urine. Or by testing sputum and stool. But by this method, by conventional method, early detection is not possible. Okay. ಈ ಮೆಥಡ್ನಿಂದ ನಾವು ತುಂಬ ಮುಂಚೆ ಕಾಯಿಲೆಯನ್ನು ಕಂಡುಹಿಡಿಯೋಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಉದಾಹರಣೆಗೆ ಕ್ಯಾನ್ಸರ್ ಬಂದಾಗ ನಾವು ಕ್ಯಾನ್ಸರ್ ಪೇಷಂಟನ್ನು ಕರ್ಕೊಂಡು ಹಾಸ್ಪಿಟ್ಲಿಗೆ ಹೋದಾಗ ಅಲ್ಲೇ 3ನೇ ಸ್ಟೇಜ್ ಇದೆ, 4ನೇ ಸ್ಟೇಜ್ ಇದೆ, ವಾಸಿ ಆಗಲ್ಲ ಅಂತ ಹೇಳ್ತಾರೆ. ಓಕೆ. ಅದೇ ರೀತಿ ಏಡ್ಸ್ ಬಂದಿರತಕ್ಕಂಥನು ಕೊನೆ ಸ್ಟೇಜ್ನಲ್ಲಿ ಹೋದಾಗ ಇವನು ಬದುಕೋದಿಲ್ಲ ಅಂತ ಹೇಳ್ತಾರೆ. ಓಕೆ. ಆದರೆ ತುಂಬ ಅರ್ಲಿಯಾಗಿ ಹೋದರೆ ಅವರನ್ನು ನಾವು ಬದುಕಿಸಬಹುದು. ಕ್ಯಾನ್ಸರ್ನಿಂದ ಅವ್ರನ್ನ ಬದುಕಿಸಬಹುದು, ಏಡ್ಸ್ನಿಂದ ಬದುಕಿಸಬಹುದು. ಅದನ್ನೇ ನಾವು ಮಾಲಿಕ್ಯುಲರ್ ಡಯಾಗ್ನಾಸಿಸ್ ಅಂತ ಕರೆಯೋದು. In molecular diagnosis, we are making use of molecule for the identification of the disease. Molecule means DNA or RNA. Okay. Now, for the early detection of the disease, some modern techniques are more useful. They are recombinant DNA technology or DNA technology, PCR technique, polymerase chain reaction technique, enzyme linked immunosorbent assay. One more is there, RIA, radio immunoassay. Okay. Now look here. In conventional method of identifying the disease or treating the disease, what happens? Presence of pathogen. Pathogen means mainly the bacteria, virus, or the fungus, etc. are suspected only when pathogen produce symptoms. So in conventional method of diagnosing the disease, the pathogen is identified. Pathogen may be bacteria or virus, suspected only after it expressed the symptoms. Okay. ನಾವು ಡಾಕ್ಟ್ರ್ ಹತ್ರ ಹೋದಾಗ ಸಿಂಪ್ಟಮ್ಸ್ ಹೇಳ್ತೀವಿ, ಕಾಯಿಲೆ ಬಗ್ಗೆ ಚಿಹ್ನೆ ಹೇಳ್ತೀವಿ. ಆಗ ಡಾಕ್ಟರು ಬ್ಲಡ್ಡು, ಯೂರಿನ್ನು ಟೆಸ್ಟ್ ಮಾಡಿ ಯಾವುದರಿಂದ ಬಂದಿದೆ, ಬ್ಯಾಕ್ಟೀರಿಯಾದಿಂದ ಬಂದಿದೆಯೋ, ವೈರಸ್ನಿಂದ ಬಂದಿದೆಯೋ, ಯಾವ ಕಾಯಿಲೆ ಅಂತ ಕಂಡುಹಿಡಿತಾರೆ. What happened by this time? By this time, the concentration of pathogen is very high in the body. Bacteria or virus multiplied in the body. The concentration is high. Okay. But in molecular diagnosis, very low concentration of bacteria or virus can be detected even before the expression of symptoms of the disease. How? By amplification of the genetic material, DNA or RNA, using PCR technique. You know the details of PCR technique. Okay, PCR ಟೆಕ್ನಿಕ್ನಿಂದ DNA ಅಥವಾ RNAಯನ್ನು ನಾವು ಉಪಯೋಗಿಸ್ಕೊಂಡು early detection of the disease is made. Okay, what are the uses of this application? Okay, PCR method is now used to detect HIV infected AIDS patient. PCR-ನಲ್ಲಿ ನಾವು ಬಹುಬೇಗ ಅಂದರೆ ಮುಂಚೆ ದ ಡೇ ಆಫ್ ಇನ್ಫೆಕ್ಷನ್ ಅಂತ ಹೇಳ್ಬೋದು, ಬೇಕಾದ್ರೆ HIV ಕಂಡುಹಿಡಿಬೋದು. ಯೂಸಿಂಗ್ PCR technique, we may detect the mutations of genes in suspected cancer patients. ಕ್ಯಾನ್ಸರು ಬಂದಿರತಕ್ಕಂಥ ಅವನ್ನ ಬೇಗನೆ ಕಂಡುಹಿಡಿಬಹುದು ಕ್ಯಾನ್ಸರ್ನ. PCR is used to identify genetic disorders. Even by using the PCR, genetic disorders may be identified. Okay. ಯಾವ ಥರ ಡಿಸೀಸ್ನ ಕಂಡುಹಿಡಿತಾರೆ ಅಂತ ನೋಡ್ರಿ. ಮೆಥಡ್ ಆರ್ ದಿ ಪ್ರೊಸೀಜರ್. ಪ್ರೊಸೀಜರ್ ಯಾವ ಥರ ಇದೆ? A single stranded DNA or RNA is tagged with radioactive molecule, what is called probe, to hybridize. Then detected by autoradiography. Mutated gene will not appear on photographic film. By this method, easily identified. ಯಾವ ಥರ ಮಾಡ್ತೀವಿ ಅಂದರೆ, ಈಗ ಡಿಸೀಸ್ ಯಾವುದರಿಂದ ಬಂದಿದೆ ಅಂತ ನೋಡಿ ಅದರ DNA ತೆಗಿತೀವಿ ಅಥವಾ RNA ತೆಗಿತೀವಿ. ಆ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNAಗೆ ಅಥವಾ RNAಗೆ ನಾವೇ ತಯಾರು ಮಾಡಿರ್ತಕ್ಕಂಥ DNA ಅಥವಾ RNA ಪ್ರೋಬನ್ನ ಲಿಂಕ್ ಮಾಡ್ತೀವಿ, ಹೈಬ್ರಿಡೈಸ್ ಆಗುತ್ತೆ. ಎಲ್ಲಿ ಹೈಬ್ರಿಡೈಸ್ ಆಗಲ್ಲ ಅದು ಜೀನು ಮ್ಯುಟೇಷನ್ ಆಗಿದೆ ಅಂತ ಗೊತ್ತಾಗುತ್ತೆ. ಗೊತ್ತಾಯ್ತಾ? ಇದನ್ನು ಆಟೋ ರೇಡಿಯೋಗ್ರಫಿ ಮಾಡಿದಾಗ ಗೊತ್ತಾಗುತ್ತೆ. ಸೊ ದಿಸ್ ಈಸ್ ಹೌ ದಿ early detection of cancer, early detection of HIV infection may be diagnosed through molecular diagnosis, that is using DNA or RNA. Okay, next, Elisa. What is this Elisa? Elisa means enzyme linked immunosorbent assay. enzyme linked immunosorbent assay. Okay. So ELISA test is based on the principle antigen and antibody interaction. Very important for CET or NEET. ELISA test is conducted to test the 8 patients. 8 patientನಲ್ಲಿ HIV-ನಲ್ಲಿ RNA ವೈರಸ್ ಇರುತ್ತೆ. ಓಕೆ, ಇಲ್ಲಿ ಆಂಟಿಜನ್ ಆಂಟಿಬಾಡಿ ಇಂಟ್ರಾಕ್ಷನ್ ಪ್ರಿನ್ಸಿಪಲ್ ಮುಖಾಂತರ ಏಡ್ಸ್ ಇನ್ಫೆಕ್ಷನನ್ನು ಕಂಡುಹಿಡಿತೀವಿ. Infection can be detected by the presence of antigens, that is proteins, glycoproteins, or detected by the antibodies synthesized against the pathogen. HIV ಇನ್ಫೆಕ್ಷನ್ ಆದಾಗ ಅದು ಫಾರಿನ್ ಬಾಡಿ ಆಗಿ ನಮ್ಮ ಸೆಲ್ಸಲ್ಲಿರುತ್ತೆ. ಇಮ್ಮಿಡಿಯೇಟಾಗಿ ನಮ್ಮ ಸೆಲ್ಸು ರೆಸ್ಪಾಂಡ್ ಮಾಡಿ ಆ್ಯಂಟಿಬಾಡೀಸ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರ್ತವೆ. ಈ ರೀತಿಯಾಗಿ ನಾವು ಡಿಸೀಸ್ನ ಬೇಗ ಕಂಡುಹಿಡಿಯಬಹುದು. ದಟ್ಸ್ ಆಲ್ ಅಬೌಟ್ ದಿ ಟಾಪಿಕ್ molecular diagnosis. Thank you. Very important for the examination. Okay.